Yes, that's no problem. I'm just going to share the PowerPoint. So before we start, I wanted to give a couple of reminders. Next week is March break. Therefore, there will be no seminar next week, and they will be returning on March 8th. We also wanted to remind everyone that the Experimental Surgery and IRR Program Joint Research Day is on May 5th, and there will soon be a call for abstracts and more information on how you can submit your abstracts if you'd like to participate. So now I'm going to pass it off to Kashif Khan for his presentation this week.
Hi, everyone. Thank you for having me here today. So yeah, my name is Kashif. I'm a now fifth-year PhD student in the experimental surgery program. I'm under the supervision of Dr. Renzo Ciceri and Dr. Adel Tritani. And yeah, I'll be presenting one of the projects from the lab. Has a mix of everything, animal studies, in vitro work, as well as some mechanistic work. So hopefully you get a full range of experiments. So this is the title. You can see my screen? Everything's okay? Okay. Yeah, so it's amniotic stromal stem cell loaded hydrogel repairs cardiac tissue in infarcted rat hearts via paracrine mediators. Okay. So after a myocardial infarction occurs, the cardiac tissue becomes irreversibly necrosed. And this is because the adult cardiomyocytes are what we call terminally differentiated, meaning they have very little or almost no ability to proliferate or repair themselves after injury. Now, the heart tries to compensate for this by undergoing left ventricular hypertrophy in order to reduce the level of stress on the ventricle. And this can be adaptive at first, but over time, it just simply isn't enough to overcome the metabolic demands of the body, thus inevitably leading to heart failure. And the unfortunate truth is that 50% of patients that are diagnosed with heart failure will die within 5 years of their diagnosis. The current treatments are mainly supportive, like lowering blood pressure or removing the excess volume, because these patients tend to be volume overloaded. But they don't really work to actually reverse the effects of heart failure or regenerate the heart tissue. And so this leads to the inevitable treatment, which are either the use of a left ventricular assist device. It's a mechanical pump that's placed within the body via open heart surgery, and that pumps for the heart itself, or a heart transplant. But those have several limitations of their own. So the question is, can stem cells be used to repair the heart? Well, this is an age-old question that has been asked for more than 20 years. And the answer is kind of. Although some studies show that they worked, they really have been shown to have poor engraftment results. This is because either the cells die after they're injected into the heart, or they're simply diffused into the circulation and they don't actually remain or engraft in the heart. And then studies that have been done in humans have shown limited improvements. So this is the biggest problem that the study I'm presenting today will try to solve. It's that the direct injection of stem cells provide limited results, but still remains a promising therapy. And so one set of next generation therapies is the use of bioactive scaffolds. And these are nanomaterials that really help with the engraftment of these cells in a tissue. There's different types of scaffolds. There's patches that, let's say, you can more like a bandage approach onto the tissue. There's microparticles, and you can put stem cells or other drugs inside these microparticles and inject it into the heart. But today I'll be speaking about hydrogels. And hydrogels, I believe, are special because they have a high water content, creating a nice nutrient-rich microenvironment for the stem cells that are injected, as well as the residing cells. They have a rapid liquid to gel transition time. So even though we inject it as a liquid, in 37 degrees temperature, it quickly gelates, creating a nice solid matrix for the tissue, thus creating a supportive environment for the cells. Other studies have shown that there are some innate regenerative effects. And one of these, for example, is the homing of immune cells to the infarcted tissue. So I think that's pretty interesting. And then finally, the use of hydrogels allows for a transendocardial delivery approach. So there's no longer a need for open heart surgery. We can deliver this therapy through a minimally invasive catheter. So the objectives of the study was to introduce a hydrogel we had created in the lab made up of chitosan and hyaluronic acid, as well as a novel stem cell source, the amniotic stroma mesenchymal stem cells that are generated from the post-term placenta, and determine their combined effects on cardiac repair. And then one of my most important roles in this project was to understand the mechanisms by which this may occur. And our hypothesis is that the hydrogel creates a supportive environment for the sustained release of extracellular vesicles from these stem cells, which contain proteins involved in regeneration. Okay, so let's get started. So in the first part of this project, we used a rat myocardial infarction model. So this is the open heart surgery we did on the rats. You can see the heart is pumping quite quickly, and we ligated the left anterior descending artery, which provides blood mainly to the left ventricle. And then we injected the stem cell hydrogel composite into the peri-infarct region, so just around the necrosed tissue. There are 4 groups. So first is the MI alone, no treatment, then the stem cells alone, the hydrogel alone, and then the composite stem cell hydrogel group as well. We used an experimental time point of 5 weeks, and then we followed up with echocardiography as well as some histological analysis. So first, we wanted to characterize the stem cells we were using. So I mentioned they're from the post-term placenta, and they are generally mesenchymal stem cells. But what's interesting about these cells is that they actually display some cardiac phenotype. So using a calcium imaging immunofluorescence assay, we were able to see some beating of these stem cells, which is similar to what cardiomyocytes do in vitro. And we also see the expression of several cardiac markers, including GATA4, a transcription factor, CircuitA, a cardiomyocyte calcium pump, connexin 43, which is a gap junction protein, and ALChem, which is a cardiac progenitor cell marker. And so our echocardiography results after 5 weeks showed improvements in functionality of the heart. So I have 3 outcomes that I've shown here. The first is the ejection fraction, which is the percentage of blood ejected from the left ventricle versus the amount of blood that remains. Fractional shortening, which is a similar measurement, but uses the diameters of the ventricle instead rather than the blood. And the stroke volume, which is more of an absolute measure of blood ejected from the ventricle. And so as you can see, at least with ejection fraction, fractional shortening, the composite scaffold and cell group show the largest improvements in function compared to the controls. And there were some independent effects seen with the groups treated with the hydrogel alone. This is indicated by the red group here and here as well. However, with stroke volume, we only see improvements in mice or rats treated with the composite group, with no changes seen for any of the individual groups. For histological analysis, we use Mason's trichome staining, and this allows us to qualitatively and quantitatively look at the level of fibrosis in the scar, in the cardiac tissue. And similar to our previous results, the composite scaffold showed about over a 20% reduction in fibrosis compared to controls, and there were some significant results seen for the mice treated with the cell group alone. We also looked at CD31 stain, which is a marker of endothelial cells. And so we counted the new microvessels that were being formed as a marker of angiogenesis. And there were no differences seen between any of the independent groups, but the composite scaffold did see quite a large effect. We looked at some other markers, so Ki67, which is a proliferation marker, and we see that the stem cells that were injected into the heart indeed did express this marker. So there's some proliferation that is happening. And these cells also maintain the expression of those cardiac markers, like I mentioned before. So myosin heavy chain 7, troponin T2, connexin 43, and GATA4 as well. So together in this first part of the study, we showed that this composite scaffold allows the stem cells to not only engraft, but also proliferate in the infarcted tissue. And this showed improvements in functionality and in cardiac function through the echocardiographic measurements I showed before. So in the second part of the study, like I mentioned before, we wanted to look at some of the mechanisms that might be taking place. And so we looked at the extracellular vesicles. So similar as to before, we took these stem cells from the human placenta. We characterized these cells under an hypoxic environment to mimic what happens in an infarct. We then isolated the extracellular vesicles from these cells in the hypoxic environment through sequential ultracentrifugation. And then we did some proteomics and pathway analyses. So first we show that even after 48 hours of hypoxia, these cells really maintained their viability with no significant apoptosis seen. And this was done through the Nexin 5 apoptosis assay. And this is likely due to the increase in hypoxia-inducible factor 1 we saw in these cells, which is a cytoprotective protein and leads to other downstream effects like increased expression of phosphorylated AKT. We also see increased expression of some pluripotency markers like SOX2 and NANOG, as well as the expression of IL-1, GATA4, troponin T, and myosin heavy chain 7. And this was all mRNA expression. And importantly as well, we also see increased nuclear localization of GATA4, which I mentioned was a transcription factor. And just a bit of background, increased nuclear localization of GATA4 means more likely an induction of cardiomyocyte differentiation. So not only are these cells, do these cells remain viable after 48 hours of hypoxia, they may be undergoing some form of a cardiomyocyte differentiation as well. So then we characterize the extracellular vesicles released from these cells. As I mentioned, we use ultracentrifugation to isolate the vesicles. We then use nanocyte tracking analysis, which tracks the Brownian motion of small particles in order to estimate their diameter. And we see that the vesicles that were released are mainly 50 to 150 nanometers in size, which are highly characteristic of exosomes. We did an flow cytometry and found expression of CD9 and CD63, which are exosome markers. And we also qualitatively looked at these vesicles under transmission electron microscopy, and we can see very small vesicles, which are indicative of exosomes. So then we did some proteomics analysis. We found that there were 122 differentially expressed proteins in the extracellular vesicles released from the stem cells under hypoxic conditions compared to those under normal or normoxic conditions. And there are a whole slew of pathways that were differentially expressed, but I've highlighted just some important ones that were relevant to the study, like angiogenesis, cell proliferation and survival, as well as extracellular matrix remodeling. So then we took this study one step further, and we tried to look at some of the mechanisms at play. So of the top 20 hits, the first one, or the one that was most differentially expressed, was SFRP1, which is secreted frizzled related protein 1. And this particular protein is part of the Wnt signaling pathway. Now, I'm a big fan of the Wnt signaling pathway, and some of you may have heard of it, but just a quick background. It's quite a complex biological signaling cascade involved in processes from proliferation, differentiation, migration, to certain disease pathogenesis like cancer, and has also been shown to play a role in embryogenesis and regeneration. So it really does a whole bunch of things. And the only thing you need to know to understand the next set of results is right here on this slide. So the pathway can be broken down into two main pathways: the canonical pathway, which is mainly signified by increased beta-catenin nuclear localization, and the non-canonical pathway, which is basically a whole set of other pathways not associated with the canonical one. And what we need to know is that if we inhibit this canonical pathway, we see differentiation of stem cells toward a cardiac lineage. And if we activate the non-canonical pathway, we see the same thing. We see differentiation towards a cardiomyocyte lineage. So in the next set of results, we did some further pathway analyses, and we found that there were several of these Wnt signaling pathways that were differentially expressed in the proteins from the extracellular vesicles that mainly belong to the non-canonical pathway. As I mentioned before, more non-canonical pathway means more cardiac differentiation. Further analysis, we found that there is increased expression or differential expression of this 26S proteasome, which was found to be involved in the degradation of beta-catenin. And as I mentioned before, if we're degrading beta-catenin or inhibiting it, we are deactivating this canonical pathway, thus further promoting cardiac differentiation. So together, these results show that there are proteins involved in these extracellular vesicles that inactivate the canonical signaling pathway and activate the non-canonical pathway together to induce differentiation of the mesenchymal stem cells towards cardiomyocytes. So that concludes my presentation. Just to sum up everything I mentioned, so in the first part of the study, we used this hydrogel stem cell composite approach and injected it into infarcted rat hearts, and we found improvements in cardiac function and morphology 5 weeks post-MI. The injected stem cells showed evidence of proliferation and cardiac differentiation, and that these regenerative effects may be in part through the sustained release of extracellular vesicle proteins, which, as I mentioned before, there were a whole set of pathways that are involved, but Wnt signaling may play a particularly important role in this mechanism. And so I just wanted to thank, of course, my supervisor, Dr. Renzo Ciceri, who's been very supportive of my work over the past 5 years, as well as other members of the lab, especially Ms. Ida Duresh, as well as members of Dr. Shortani's lab and Dr. Rack's lab that have been integral to the success of this project. So thank you, and I'd be happy to answer any questions.
Kashif, that's really wonderful work. So the first part, when you inject the stem cells and then you look at the markers. What you're looking at is in situ dependent differentiation.
That's right. Yeah.
Okay. So how would you know that those cells that you injected, they are not the native cells? So how do you know you're not looking at native cells as well?
Yeah, maybe I skipped over that, but these are actually human amniotic stromal mesenchymal stem cells that we injected into a rat model. So we used a human mitochondrial protein to identify the cells first, and all the other antibodies we used were also human proteins. So we know that we're identifying specifically these cells and not any of the resident cells.
Oh, that's great. Thanks.
Mm-hmm. Yeah, it's interesting. These cells, even though they're of human origin, don't really show any immunogenic response, and other studies have shown similar results as well.
Okay, thanks.
So thank you, Kashif, for a great presentation. I just want to ask you, did you ever try the secretome versus the whole secretome versus the extracellular vesicles? Why did you home in on extracellular vesicles as opposed to the whole secretome?
Yeah, that's a great point. It's likely that we were to see perhaps even greater effects with the whole secretome, but I want to be very specific, I guess, in what I was using. And a lot of the studies that are done today have been focusing on these vesicles as well, because we don't really know everything that's in the secretome. Some could be, for example, could be immunogenic, could be harmful to the native tissue. And so for me, I have a special interest in the vesicles. That being said, Ida, who's in our lab, is focusing more on the secretome analysis. So she'll be continuing this work, but more from the secretome perspective.
Thank you.
Maybe I can ask a question if that's
yeah, sure, Doctor.
So in your proteomics study, how much garbage came out of the analysis? Did you have a lot of proteins that appeared non-specific, and what strategy did you use to focus on the targets that you focused on?
Yeah, and absolutely, there was a lot of garbage that came with it. So in collaboration with the proteomics platform at the RI, we set a cutoff limit. So I think it was five hits for.
Okay, number of peptides was one criteria. Okay, that's good.
Okay. So with that, we were more confident in the positive in our positive results.
Very impressive study and very good talk. Congratulations.
And could you also tell us about the hypoxic condition that you used? That I thought that was very interesting. What kind of hypoxia? How? What's the percent oxygen tension, and also how long?
Sorry, Dr. Philip, I might have missed that. Can you repeat
the hypoxic preconditioning?
Oh, yes.
Hypoxia, what are the conditions that you used? How much oxygen tension, and also how long, the duration?
Yeah, so it was a hypoxic chamber that was used in collaboration with Dr. Rack's lab. So they have a whole tissue culture hood specified for that. And it was a 0% oxygen for 48 hours. Other models that we've used in the lab, you can, if anyone else is interested in hypoxic studies, you can purchase these small hypoxia chambers for smaller cell culture work.
Okay. Thank you. Okay, I asked that question just because the graded hypoxia would have differential effects. 0% oxygen versus say 5%. The response, cellular response could be very different.
Yes, absolutely. And that was mainly done because of what other studies have done as well. And we also want to mimic what happens in an infarcted microenvironment, which is the necrosis around the cells. It really is almost 0% oxygen. And that's why the cardiomyocytes die. So we really want to mimic what happens clinically.
Yeah. Okay, thank you.
Yeah, no, that was a wonderful talk, actually. I was kind of very much impressed with your data. Just have one question, though. So when you're culturing the cells in the hypoxic condition, what type of culture media did you use? Did you use like a differentiation media or something to induce, or just?
Nope, it was a regular culture media. So the differentiation effects that we saw were independently from the hypoxic conditioning. We didn't change the media at all. Sorry, I should clarify, we removed any serum or glucose, but the basal media remained the same.
Correct, thank you.
All right, is there any last questions before we move on to the guest speaker, Dr. St-Arnaud? Okay, that seems to be the last of the questions. Thank you so much, Kashif, for your great presentation again. Now I will be passing the baton off to Dr. Annie Philip, who will be shortly introducing Dr. St-Arnaud.
Okay. So I'll go on to do just that. Okay. Yeah. So it's my pleasure to introduce and welcome Dr. St-Arnaud, today's faculty speaker. So his talk is entitled "A Small Molecule Lead Compound Blocking FIAT Activity Increases Osteogenesis." Dr. St-Arnaud is the inaugural Francis Gloria Professor in Pediatric Musculoskeletal Research at McGill University. He's currently the Director of Research at St-Arnaud Hospital in Montreal. He holds cross-appointment as a tenured professor of medicine, surgery, and human genetics at McGill. His research efforts are focused on vitamin D metabolism, as well as the control of gene expression in bone-forming cells. His contributions to the understanding of cell differentiation, bone cell differentiation and function have been recognized through several distinctions, most recently the C.P. Leblon Career Award. Since the beginning of his career as an independent scientist, Dr. St-Arnaud has supervised more than 50 graduate students and postdoctoral trainees. He has a total of 150 publications with several book chapters and also conference proceedings. Dr. St-Arnaud also holds several intellectual property rights. We are delighted to have you, Dr. St-Arnaud, and we welcome you again and look forward to your presentation, Dr. St-Arnaud.
Thank you very much. Thank you for the invitation. I'm very happy to be able to share the most recent progress on a project that we started way back when, and for which we have very exciting results. I'm following a great presentation. I hope I'll be up to the task. So the title is a small molecule lead compound, which means we're going to foray into drug discovery. And the topic is about bone tissue. But even if you're not familiar with the tissue or with the genes that are expressed there, I hope you'll be able to follow the pathway that is normally used for drug development. I disclosed the fact that some of the intellectual property that was used to screen for identifying the lead compound are patented, and I'm the named inventor on that patent. And before I forget, I tried to give credit where credit's due. The project dates back 25 years. We cloned the target in 1997. And those were the people in the lab at the time. Gurgen cloned the first partial cDNA of the target. Louise and Mia are still working for us as animal health technicians. And Alice retired just last December after 28 years in my lab. Live cloned the full-length cDNA of the target. Vianney did the transgenic mouse. Bahari did the knockout. And the current student working on the project and the most recent data I'm going to present are from Caitlin Anderson, a PhD student registered in human genetics. Caitlin is on the Zoom conference, so I hope I'll do credit to her data. Some of the initial data was supported by an NIH grant, which I forgot to acknowledge here with the logos. After that, the screen for the lead compound was supported by a developmental grant by Shriners Hospital for Children, and we leveraged that into a full 5-year CIHR grant that we currently hold. So this is the drug development process, the discovery phase and the development phase. And I'm going to take you from here up to about here. Very excited of the progress we've made over the years. So the first step is know what it is that you want to address, what it is that you're interested in. We work in a pediatric hospital that is specialized in bone disease and orthopedic issues. And so the main patient population are children with osteogenesis imperfecta, which have fragile bone with very low bone mass. So we would be interested to find treatments that could increase the amount of bone that these children have. Right now, the current standard of care for this disease are anti-resorptive agents, so we can block the loss of bone, but there's no approved therapy yet to be able to replace lost bone tissue and allow a gain of bone mass. There are, in fact, 3 approved therapies to stimulate bone formation in adults. One of them is based on parathyroid hormone, the first 34 amino acids. The compound under development was called teriparatide, and the drug is commercialized under the name Forteo. There is an equivalent for the PTH-related protein, and that was abaloparatide, which is now commercialized as Timlos. And there is one humanized antibody that recognizes an inhibitor of bone formation. So it blocks an inhibitor which stimulates bone formation. The name of the molecule under development was romosozumab, and it's now commercialized under the brand name Evenity. All of these have some limitations. 2 of them, Timlos and Evenity, can only be used for 2 years because there are risks of serious side effects, either bone cancers or cardiac issues. The main drawback for Forteo is the daily injections. It's the same treatment as for insulin for diabetic patients. And in the case of bone diseases, there's very low compliance. After a few months, a patient quit the therapy. And also in our case, we work at a pediatric hospital. None of these treatments are currently approved for children. So what is really lacking is an efficacious orally administered bone anabolic therapy, and this remains an unmet medical need. And we are challenged to find alternative treatments than the ones that are available. One strategy that we used in the lab was to look at the pathways operating downstream from PTH interaction with its receptor, and this work is in progress in the lab. But also some of the specialty in my laboratory is to identify targets that can be druggable to be able to impact the tissue. And this is the type of strategy that I'm going to describe right now. So this is the disease. We want to increase bone formation. We want a bone anabolic compound. So we need a target and to try to characterize it in a way that says, would it be a good target and would it be druggable? And so when we initially cloned the target, we didn't know what it was doing. It was, we cloned this in a protein-protein interaction assay with another transcription factor that we were interested in the lab. And we're still working on that aspect of the interaction. But what struck us when we sequenced the clone that we had in hand was the fact that it contains a protein motif that is called the leucine zipper, which is a protein-protein interaction motif. It's a rather simple motif. It has no strong sequence similarity. It is only leucine residues spaced every 7 residue in the protein. And you can see that there are 3 such leucine zipper motifs in the cDNA that we clone. So right away, well, I'm going to give you a little bit more information about the leucine zipper. It's really when the helix forms and the leucine residues align all along one face of the helix, because 7 residues is the number of residues it takes to make 1 turn of alpha helix. And it is truly a protein-protein interaction motif. In transcription factors, it brings the 2 members of the dimer together, and then it positions a basic domain that is able to bind DNA. And this is how transcription factors interact with their cognate recognition element and regulate transcription. So you can think of this part of the protein, the leucine zipper, as really a module necessary for protein-protein interaction, a little bit like the Velcro type interaction. And so we saw this motif in the clone that we obtained, and the question we asked right away is, can it homodimerize, or can it dimerize with something else? Can it interact with other proteins that have a leucine zipper? And so we reused the protein-protein interaction assay, which at the time was the yeast to hybrid assay. And we tried different combinations. We made a bait with the target that we have, and we made a target with the bait that we have to see, can it homodimerize? And when the bait and the target interact in the yeast to hybrid assay, the yeast can grow on selection media. And so these 2 fields here, where you can grow yeast when there's no selection, but when you put the selection, these yeast do not grow there. This is the bait with the target, and the same with a little bit of a mutation, a partial cDNA. So this result told us the target that we have cannot homodimerize. But when we screened at large a library of cDNAs from bone cells, we ended up with 3 independent clones of the transcription factor ATF4. And this is what we see here. This is the target as the bait with an ATF4 target, and yeast grow. And this is the positive control in the assay. And we can confirm these interaction data from the yeast system with endogenous proteins in the cells. If we immunoprecipitate ATF4, the target that we had identified, to which we gave the name FIAT, and I'll explain the acronym in a moment, co-precipitates with ATF4, and we can do the reciprocal immunoprecipitation. If we immunoprecipitate FIAT, ATF4 comes with it. And these are endogenous proteins in the osteoblast. These are not overexpressed proteins, so we can see that this interaction is at least relevant in osteoblast, in bone cells, in tissue culture. And we were very, very excited when we found this interaction, because soon after we got this result, in the literature appeared a number of studies that showed that ATF4 is critical for a number of functions in bone cells. First of all, it was first cloned as a regulator of the transcription of genes involved in amino acid import in cells. And this is very important in bone cells because they have an extensive matrix formation of type 1 collagen. And if you modulate the amount of ATF4 in osteoblast, you have an impact on how much type 1 collagen it can synthesize. ATF4 in osteoblast also controls the onset of osteoblast differentiation. It is coupled to resorption because it regulates the expression of a cytokine that regulates osteoclast differentiation. And it's also involved in the control of energy metabolism from the skeleton through various mechanisms of action. So you can see it's a critical regulator of osteoblast biology, and we had found a protein that interacts with it. You can understand that with such an array of functions, the activity of ATF4 is tightly controlled in bone cells. One first mechanism of control is through its half-life. Strangely enough, if you look at ATF4 expression, you will find mRNA virtually everywhere in every tissue, but the protein seems to accumulate only in osteoblast at any measurable level under steady state. And that's controlled through ubiquitination and proteasomal degradation. Its activity as a transcription factor, where it will bind and which genes it will activate, is controlled by post-translational modifications. It can be acetylated and it can be phosphorylated by 2 different kinases on 2 different serine residues, and that will control which partners it interacts with and which genes it controls. But also there's another level of control through the leucine zipper, because ATF4 is a leucine zipper protein. It can homodimerize, but it can also dimerize with other partners. And this will change the array of genes that it will control. It will change the binding site with which it can interact in DNA. But there are also inhibitory interactions. And one of those inhibitory factors is the target that we cloned, to which we gave the name FIAT, which stands for Factor Inhibiting ATF4-Mediated Transcription. I come back to the leucine zipper. I've mentioned that once the 2 interaction domains align with one another, it brings the basic domain of the transcription factor in the right conformation to interact with DNA. And this is seen with the canonical homodimers or the heterodimers of the AP1 or ATF4 family, or ATF family. But the protein that we cloned, FIAT, has a leucine, in fact, has 3 different leucine zippers, but it does not have a basic domain like that. So this would suggest that, in fact, it forms inactive dimers. It will dimerize with ATF4, but because it cannot form the DNA binding domain, this dimer would not be able to bind DNA. And this is, in fact, what we observed. This is a canonical ATF4 probe that is labeled with radioactivity and that migrates in the gel. This is an electrophoretic mobility shift assay to look at protein DNA interactions. We use a nuclear extract from osteoblast. There's a number of proteins that bind the probe, but if we use an anti-ATF4, we can see that it is this band that disappears. So this represents ATF4 binding to the probe. And when we increase the amount of recombinant FIAT in the assay, we get the same effect as the antibody that blocks ATF4 function. We see that ATF4 is no more able to bind to its binding site. So it cannot bind DNA, and because of that, it cannot activate transcription. These are 2 different reporters. This is a canonical ATF reporter. When we overexpress ATF4, you can see the response in terms of transcription. FIAT does nothing on this reporter because FIAT cannot bind DNA. But if we put the 2 together, we have a significant inhibition of ATF4 activity. This was with a multimer of the canonical ATF binding site if we take the binding site from the gene osteocalcin, the name of the gene is BGLAP. This is a marker of osteoblastic differentiation and the first bone-specific target identified for ATF4. You can see that ATF4 activates the expression from the reporter. FIAT does nothing, but when you put the two together, FIAT in a dose-dependent manner inhibits the activity of ATF4 on the osteocalcin promoter. I mentioned before that the activity of ATF4 can be stimulated by different protein kinases. This is in the presence of RSK2. You can see that the activity of ATF4 is potentiated by phosphorylation. But when you put FIAT together, again, you have a strong dose-dependent inhibition of ATF4. So this really highlights the biochemical activity of FIAT. It interacts with ATF4 and it prevents its activity as a transcription factor. I mentioned that that FIAT has 3 different leucine zippers. We were interested in finding out which one mediated the interaction with ATF4. So we mutated each one of them in turn. We mutated the leucines for alanines. And when we do that with the first leucine zipper, there's no effect. The protein still inhibits ATF4 activity. If we mutate the third zipper, again, no effect, it still inhibits. But when we mutate the second zipper, the one in the middle, this is when the interaction is broken, and this mutant cannot influence ATF4 transcriptional activity. This is with a site-specific mutant. If we delete the mutant, the protein is a little bit shorter, and we see the same effect. So it's the second leucine zipper that mediates the activity or the interaction of FIAT with ATF4. At the level of osteoblast activity, this is a culture of osteoblastic cells that have mineralized their matrix. In the tissue culture dish, the brown stain is the deposited mineral. If we overexpress the wild type FIAT in that context, we see that we prevent mineralization. So we block the activity of ATF4 to induce osteoblastic differentiation and activity. But if we overexpress the protein with the deleted zipper 2, then the cells can mineralize again. So both at the level of gene transcription, but also osteoblast function, the activity is mediated through the second leucine zipper. So this is data obtained by overexpressing the FIAT target. What happens if we remove FIAT from osteoblast? So in this case, we used a shRNA-mediated knockdown, and at the RNA level, we were highly successful, at the protein level as well. So now osteoblasts do have only a very low residual level of FIAT protein in them, and under those circumstances, the expression of the osteocalcin reporter is dramatically increased, as well as the mineralization is enhanced in those cells. So we get reciprocal effect of overexpression or inhibition in tissue culture cells. So that was quite interesting in terms of the biochemistry and the cellular biology of the target. But does it have a physiological relevance? Does FIAT control bone mass? That's really what we were interested in before we launched into any other before we even asked the question, is it druggable? Does it in fact control bone mass? So we used 2 different strategies based on mouse molecular genetics to do this. First, we did a transgenic, which was the easy thing to do. So we put the full-length FIAT cDNA under the control of the 2.3 kb type 1 collagen promoter. This is the alpha-1 chain of the type 1 collagen molecule. And the transgene is specifically expressed in bone, not in the other tissues that we tested. We tested skin and we tested tendons, which are also type 1 collagen expressing tissues. But with this shorter fragment of the promoter, we can dramatically enrich the expression to bone tissue. The amount of overexpression that we obtain is relatively low. when we do the ratio of FIAT to endogenous gene marker, we estimate that we achieved only a 20% overexpression of FIAT. But of course, not only is it a little bit overexpressed, but it's constitutively expressed. There is no regulation of the expression of the transgene. And the impact of the transgene is really to decrease bone mass. So at the top, we have the wild type mice. This is a sagittal section in the tibia of the mice, two different stains, the Goldner stain, which stains the bone in green, and the Boncasse stain, which stains the bone in black. And this is the cortex of the tibia. And at the center, you have the bone trabeculae, or the spongy bone. Again, it's the green coloration that you should focus on. This is the bone marrow. And I think you can appreciate that in the transgenic, the cortices are thinner. We see the same with the two types of coloration, and the amount of bone trabeculae is also greatly diminished. So there is a decrease in trabecular and cortical bone mass. Of course, if you see less bone, it could be due to less formation or more resorption. So we tested that. There's no change in the markers of resorption. And we see, although we see no change in the number of bone forming cells, we can see that their activity is decreased, the mineral apposition rate and the bone formation rate are decreased in those transgenic mice. We also did some basic gene expression monitoring, especially for the osteocalcin gene, which was the first ATF4 target characterized. And you can see that the transgene inhibits the expression of osteocalcin. So when we have more FIAT, we have less bone. What happens when we have less FIAT? So we did a knockout of FIAT, and you can see that this leads to an increase in trabecular bone as well as an increase in cortical bone. And we can quantify this increase, and it is the bone volume and the thickness of the trabeculae is increased. At the level of the cortex, the increase is slight but statistically significant, and it is even more significant in the sense that it contributes to better biomechanical properties of the bone. The bone is stiffer and it takes more energy to break it. So when there is less FIAT, you have more bone and it's better quality bone. Again, this phenotype is due to an impact on the bone forming cell, the osteoblast, because the markers of bone resorption are not changed in these mice, but the mineral apposition rate and the bone formation rate are increased. So it's increased activity of the osteoblast and not a change in the osteoclast activity. Again, when we have as a readout the expression of osteocalcin, in this time when we release ATF4 from the inhibitory activity of FIAT, you get increased expression of osteocalcin. So even in tissue culture, in cultured cells, and in mice, there are reciprocal effects of increasing or decreasing FIAT expression that impact osteoblast activity and bone mass. So we concluded that FIAT is a valid target, and we hypothesized that if we could identify compounds blocking the interaction of FIAT with ATF4, this would increase osteoblast activity, and it could improve both trabecular and cortical bone mass. And at the moment, there's only one compound that can do this. It's the Evinity Antisclerostone antibody, which I remind you can only be used for 2 years and requires not daily injections, but injections at intervals. So we now embark another step of drug discovery, find what is called a lead compound that could become a drug. And so here we took advantage really of the main structure, or the main features of the FIAT ATF4 interaction, which is that their interaction is mediated by the leucine zipper. And transcription factors are modular. You know, they require a domain to bind DNA, and they require a domain to activate transcription. And we can, if we reconstitute those through a protein-protein interaction motif, you reconstitute an active transcription factor. And to have a screen assay that has very low background level, we used a yeast DNA binding domain, the GAL4 DNA binding domain. So this is not expressed in mammalian cells, so it gives very low background. And we used a viral protein, the activation domain from a viral protein, which is also not present in mammalian cells. And we put the one zipper from ATF4 in one of the components of the dimer, and the zipper of FIAT on the other one. I would need to look at my notes to remember which was which. But anyways, this was the strategy. And we put these constructs into mammalian cells now. So instead of the yeast 2 hybrid system, we have a mammalian 2 hybrid system. The canonical system used firefly luciferase, where Caitlin changed this gene for the Metridia luciferase, which is secreted, and it made it easier to have a high throughput assay where we did not have to break the cells, but we could just harvest the supernatant from the cultures. And Caitlin prepared 3 cell types, negative control cells that were transfected with the reporter only. As I say, there is no GAL4 in mammalian cells, so the background was extremely low. There was one clone transfected with the reporter, but also a fusion, a direct fusion of GAL4 with BP16. And this was highly active and led to very high levels of luciferase expression. And then the mammalian 2 hybrid, in which we had the bait and the target, which interacted through the FIAT and ATF4 leucine zippers. And we were pleasantly surprised that this mammalian 2 hybrid system had a dynamic range that was almost identical to the direct fusion of the 2 parts of the transcription factor. So very high dynamic range in the assay. We contracted out to our partner at the Université de Montréal, the Institut de Recherche en Immunologie et Cancer. The IRIC has a library of 125,000 compounds and a robotic platform that we could use as a fee-for-service platform. And we screened using this mammalian 2 hybrid. And what we were looking for really are compounds that would decrease the luciferase activity, right? Because we want to block, we want to block the interaction of FIAT with ATF4. And as we screened 125,000, we ended up with 19 compounds at the initial screen. And we did those responses and focused on the 8 more promising ones. And then we threw them on osteoblastic cells. And this is the amount of mineralization that you see in cells treated with the MSO, which is the vehicle to dissolve all of these compounds. Untreated cells have similar amount of mineralization. And again, I remind you that we're trying to inhibit an inhibitor, right? And so if the compound is active, it will break the interaction of ATF4 with an inhibitor, and it will stimulate ATF4 activity. So it should stimulate osteoblastic activity and function. And this is what we see with compound C73. This is a code name. We're still working to protect the intellectual property of this finding. You can see a dramatic increase in the activity, in the mineralization activity of the osteoblastic cells. There was another interesting compound in this initial test, but we were never able to reproduce this data in future assays. So really, out of 125,000 that were screened, we ended up with 1 stroke of luck, but at least we need only 1. So we can move from there. Mineralization is one end result. What about gene expression monitoring? Can we see changes in the osteoblastic differentiation markers in those cultures? And indeed we can. All of the early markers of osteoblast differentiation are significantly increased. Genes that control the function, the synthesis of the matrix, as well as the control of active inorganic phosphate for mineralization. And finally, the initial target characterized for ATF4 in osteoblast, and this is a dramatic increase of about 5 to 6 fold. So this was done with the MC3T3 E1 osteoblast cell line, which is a cell line that was established in the early 80s. So it's important to replicate this data using primary cultures of osteoblasts. And we've done this with a primary mouse osteoblast, and you can see that the compound is very, very active in primary cells. And again, the osteoblastic markers are increased. And when we do this with human cells, which is an important control, we can stimulate mineralization and again, expression of differentiation markers. So far, we were very excited with these results in vitro, in tissue culture. And we also wanted to validate that the mechanism of action of the compound was what we thought it was, right? We didn't want something that acted through a different mechanism than what we had envisaged. So if you remember, we identified a compound that we believe disrupts the interaction of ATF4 with FIAT. So in the presence of the compound, ATF4 is released from its inhibitor, and therefore there should be more ATF4 available to bind DNA. And so we did a chromatin immunoprecipitation assay on the osteocalcin promoter, immunoprecipitating ATF4. And you can see that, in fact, when we treat the cells with compound C73, the amount of ATF4 at the proximal promoter of the osteocalcin gene is highly significantly increased. So this is proof that in the target cells, the compound acts through the mechanism of action that we had hypothesized. We have additional data with in silico assays, but I think this one really supports the hypothesis that we have put forward. So we now have identified the lead compound that hopefully could become a drug, and now we have to test it in vivo to see, is it safe, is it tolerated in preclinical models, and after that, is it efficacious in when we treat animals with it. There's a phase of optimizing the structure of the compound, but we are doing first the pharmacokinetic and the pharmacodynamic studies, but we have data on the safety and preclinical efficacy that I'm excited to share with you. So we did a dose ranging study starting very, very low. We had no idea if the compound would be well tolerated in animals. And so we increased by a factor of 10 at each experiment. We proceeded very, very cautiously, had a cohort treated with this low dose. And when the results came in and the animals tolerated well, we increased 10 times, et cetera, et cetera. And up to 10 mg per kg, there was no issues for kidney function and liver function in either males or females. Also, heart liver function was maintained. The blood biochemical markers were maintained within the normal parameters. If we looked at total blood counts in these animals, again, no impact. When we switched to these higher doses, we were surprised to see that it was below the normal range that was given to us by the laboratory who did those measurements. But when we measured animals from the same cohort with no treatment, we saw that they were highly comparable in both males and females. So what this means essentially is that the normal range that is on the considered, the range that is considered normal should in fact be adapted depending on the strain of mice that you are working with. But we conclude from these dose ranging experiments that the animals can easily tolerate 10 mg per kg daily, and we therefore moved on to preclinical testing. And there's a number of assays that we are interested in doing. I'm going to present the first data that we had, just a steady state treatment of wild type mice with just C57 black 6 mice that we purchased from Charles River and that we tested. We injected them 5 days a week, trying to give a little bit of a break to the animal health technicians over the weekends for 6 weeks. And when we do that, lo and behold, we can find that the markers of osteoblastic differentiation are all increased, whether we look at early markers like RUNX2 and SB7, or genes that are important for osteoblast function, alkaline phosphatase and type 1 collagen, as well as the canonical target of ATF4 activity, osteocalcin. All of these are significantly increased in animals that received the compound. We tested to see if there was an effect on bone resorption. Rankligin is a cytokine involved in osteoclast differentiation and function. We know it's an ATF4 target, so it is increased, but its inhibitor osteoprotein is also increased in a way that there's no change in the Rankel to OPG ratio, and we see no evidence of increased bone resorption when we look at serum markers. Interestingly, ATF4 itself is increased. We don't know the mechanism for that. Maybe ATF4 regulates its own expression under certain conditions, but FIAT itself is not involved. So that was very exciting data. This is from mRNA from the long bones of these animals, and we got the best results in female animals, but we got similar data in males. But I'm reporting only to you the results in females in the interest of time. When we look at cortical bone, we see increases in the total area and total tissue volume, not enough to get an impact on the total cortical bone mass, but still increases in the right direction. When we look at trabecular bone, here we got an increase in tissue volume and bone volume that is enough to generate a significant increase in bone mass. The bone volume over tissue volume is the index of trabecular bone mass, and also the thickness of individual trabeculae is increased in the animals that were treated with the compound. I just realized that I should have moved the slide upward. The slide, there's no dramatic impact on cortical bone, but what we see appears to be significant because it does have an impact on the biomechanical properties of the bone. It takes more energy to break the bones from the animals that were treated with C73. Right now, we're pursuing this in different preclinical models of disease. So we have a prevention strategy for osteoporosis, a treatment strategy for osteoporosis. So the model that we use is, of course, ovariectomy-induced bone loss, which is a preclinical model of postmenopausal osteoporosis. And the samples have been collected. We have impacts on bone biomechanics, which we were able to measure right away, but we are still analyzing all of the data. We also have a model that we tested for glucocorticoid-induced osteoporosis, and the samples are collected, but the analysis is ongoing. In summary, though, I think I presented you data that all of our preclinical work that we started in the late 90s support the importance of FIAT in bone physiology. And I think with all of the data, some of it I did not have the time to show, I think we have validated FIAT as a target for drug development. We had in the patent described how we could use the FIAT structure to screen in a high throughput assay to find the type of compounds that we just identified. So this was patented in the current patent on FIAT that we have. And so it was the first application of this technology for drug development. As I said, we have the patent. Now the compound itself is a new discovery, and we're hoping to be able to protect that intellectual property as well. We have identified the lead compound that enhances osteoblast differentiation and function in vitro. The compound is well tolerated, up to 10 milligrams per kilogram body weight. These are when you do these types of study initially, you use intraperitoneal injection, which is the norm in these types of studies. The next step for us will be to determine if the compound is active when it is administered orally. This may require some chemistry to change the structure of the compound, but starting from the lead compound, we can optimize the chemistry to achieve these results. The compound induces an increase in osteoblastogenesis in vivo, which is very, very exciting. Right now we are entering what is known as the Valley of 1,000 deaths, because there's only one compound out of 1,000 that are found in the pharmaceutical industry that makes it to market. But for us, it's still an exciting project that we're very excited about because drugs that increase osteoblast activity would be advantageous in the type of disease that we see at the hospital, osteogenesis and perfecta treatment, and other types of surgery. But also it does not escape us, even if we work in the pediatric domain, that it would be perhaps interesting for the treatment of adult metabolic bone disease like osteoporosis. In closing, I'd like to show this young man who's a patient of the hospital. He was born without the right arm and without the right leg. I've had the chance of going to a hockey game with him a few years ago, and when the Habs were winning a few games, but now they're back on the winning track. But it was a great experience to share with him. And Jeffrey told me, I don't consider me disabled. I consider myself differently able. And when you hear something like that, it's so inspirational that when I receive a grant that is not funded, or when an experiment in the lab doesn't work, we just get back at it and find a solution, and not get affected by despair or a gloomy mood. A great source of inspiration are our patients. And on this, I will end and gladly take any questions that you may have.
Okay, we are now open for questions. Thank you very much, Dr. St-Arnaud, for a highly informative talk. I'm sure students have several questions. Perhaps I can, as it starts coming into the chat, I can, or you can unmute yourself, of course, and ask your questions also. I can just start by asking you, could you tell us whether this compound C73 would bind to any other protein other than FIAT?
Well, we're doing those assays at the moment. We of course are targeting other leucine zipper proteins, and the data so far says that it does not disrupt Phos-Jun interaction, Phos-ATF4 interaction, or Jun-ATF4 interaction. So it really seems to have some specificity towards FIAT.
Yeah, and also I don't know whether you might have mentioned it, but perhaps I didn't get it, whether FIAT interacts with any other transcription factor other than ATF4.
That we have not tested yet, so that's something that we would.
No, I was just.
No, but our hypothesis is that it really targets the leucine zipper, because this is what we use to screen for it, and so we focus on transcription factors that have this protein-protein interaction motif. Yes.
So there you get the specificity. Even if it interacts with other transcription factors, it may not, because you are targeting the leucine zipper interacting domain. So that leads me to the other question, which is to ask that since you know so much about the structure itself, I was just wondering why you would not go the other way, like rational structure-based design of the drug, like interacting peptide or something like that?
It's a good question. There's no other reason apart from the fact that it was easy to set up an high throughput screen, and we did it. I mean, that's well, easy is a relative term, but what we were comfortable working with were really the structure or the motifs that we knew about, and that's what we focused on. But the other approach could have certainly yielded interesting data, especially if it's an artificial intelligence-based iterative model. We could have ended up with very different molecules. You're right.
Yeah, very informative.
It's a type of thing that our specialty is identifying targets. It was really our first foray into drug development per se, and therefore we come at it with not with the full mindset of the pharmaceutical industry who puts all of the possible strategies in parallel to move a project like that forward.
Okay, thank you. I'm not prying into the nature of C73, but would you like to tell us about the nature of the library that you used? Is it marine-based? Nature of the library itself, or the origin of the library?
Well, University of Montreal, when the companies left Montreal, when Merck left and GlaxoSmithKline and all, the U of M purchased a large part of their libraries and also acquired commercially available ones, and hired the medicinal chemists from the pharma industry that were in Montreal, and they are developing their own additional libraries of compounds. So there was the screen involved all of that. Unfortunately, unfortunately or not, the compounds that have been newly synthesized at U of M are protected, and we would have had to enter into an agreement with them to pursue the project. But fortunately for us, the compound that is active is just an off-patent compound that we can for which we can protect the current use that no one else has ever identified.
Thank you. So we are waiting for the students to unmute themselves. Oh, yeah, Dr. Chevalier has a question. Yes.
Yes. Thank you for the very excellent talk, and I think that for the students who are there, it's important to say that something that started years ago, if it can take 20 years to get to it or more. So I think it's an important lesson for the students. My questions are, how do you manage to present the data on C73 by students? Now that you have a lead compound, to protect the intellectual property. I think this is important for students to hear about how we can work on a project where you have a compound that is protected. So how do they present? That's my first question. And the second is the way that you think this drug could be administered. It's by injection, by pills, or the type of, it's not going to tell us what is the drug, but what would be the way to administer when you're going to study some mice or what you're doing right now?
Yeah, very, very good question. So we are hoping that either the compound in its current structure or a derivative of the compound would be orally available. That's really what we're after. The unmet medical need is an orally active compound, and we don't know that yet. But before we embark on gavage studies, we wanted to know that it was active. And to answer your other question, it's a risk what we're doing. We could have kept everything secret. My understanding is what is really new that we can truly protect is the chemical structure of the compound. So until we disclose that publicly, we can still protect it. And so right now, Caitlin has presented part of this data, and I have myself, but never disclosing the actual chemical structure of the compound. After
Okay.
that, normally I think at this stage, if I was in a regular McGill department, well, I am in a regular McGill department, but my main, well, my other affiliation is at St-Arnaud Hospital for Children, and we are bound by our policies to protect the intellectual property through the St-Arnaud hospitals. We have submitted the medical discovery literature, but the system is much slower than what would have happened at McGill. So if I had submitted through McGill channels, I'm sure that we would have a provisional patent at this time. The Shrine is very cautious about investing money because they have a history of protecting inventions that didn't go nowhere. And so they want proof that it would be active in a model of disease, which we are developing now, but
Yes.
we don't have that data. So as soon as the data comes in from the disease models, I will put pressure on the legal department at St-Arnaud Hospital to try to file for intellectual property. The rules that we've always operated on is that if they decline the first right to the intellectual property, then I'm allowed to contact McGill and to protect it through the McGill channels.
Okay. Thank you. Thank you.
Pleasure. Thank you.
And good luck. Good luck for this.
Thank you.
Any other questions? Anyone else?
Sure, yeah, I have a question.
Yes.
Great presentations and congratulations on your success. I mean, wonderful data, wonderful compound. I was just curious about FIAT and its regulation and calcium and phosphate levels, if that has any control on that. And also, have you ever tried doing a combination of, let's say, vitamin D or some calcium supplementation and the inhibitor for FIAT to see if that improves bone formation?
So we have not done those combination therapy attempts. Thank you for the suggestion. When we characterized the phenotype of the FIAT transgenic or the FIAT knockout, there was no impact on calcium or phosphate homeostasis. So we don't expect it to work through these mechanisms. We really think it's through the regulation of ATF4 activity. So maybe we could have impact on type 1 collagen synthesis, but we have not looked at that. Well, one indirect measurement is bone quality. So we have indirect measurements through bone indentation, and so there is an impact on ameliorating bone quality. But we don't think there's an impact with traditional hormonal control of bone mass.
If there are no other questions, thank you again, Dr. St-Arnaud, for a highly informative, and I'm sure it's highly inspirational for the students that research of many, many, many years actually lead to something, something tangible. And what they are doing is highly significant. It all adds up, and that it will be somehow result in some discoveries, if not from your own lab, other labs which would pick up on it. So yeah. This is highly encouraging. Thank you.
Well, thank you, and I hope it was of interest to the students.
Oh, for sure.
Thank you very much for the invitation.
Thank you.
Good luck to everyone. Bye-bye.
Bye.
Bye-bye.